It's a pleasure to be here today and to talk about our work on chromatin accessibility, transcription factor activity on single cells. And this work is about actually an analysis of single cell toxic data, which can be obtained among others, for example, by using droplet-based protocols as provided by 10x Genomics, I show here. And the readout of these experiments are going to be actually the reads, right, where we're going to have both the barcodes, which tell us which cells these reads are coming from, and the reads which are associated to the DNA regions, which now are your open chromatin regions. So you can use now this data, for example, to estimate open chromatin matrix. And this open chromatin matrix will look like this, where we're going to have how many reads you're going to have per cell and per open chromatin region. You can then use this, for example, to perform clustering, and this will allow you to find the cell types which you have in these experiments. And you can then further process this data, for example, by combining all the toxic fragments that you have in one particular cluster to create, for example, what we call pseudo-block toxic libraries. And these you can go and visualize, for example, how regulatory regions or promoter regions are open in one particular gene. Or we can also do all the standard analysis, as for example, footprinting analysis, which can help you to indicate the transcription factor activity in the different cells that you have identified. Actually, the first part of the talk is going to focus on this open chromatin matrix, right? So usually the way that you can obtain these, you can combine your toxic library as one. You can do, for example, p-calling, and this will be used to represent the regions which you have open chromatin in the cells. And then you go back to the barcodes, and then you can tell how many reads you're going to have per cell, right? And because, of course, you only have 2 chromosome copies, and given that these regions are usually small, at maximum you should have 2 reads in one particular entry here. And actually, this matrix is has very interesting characteristics. One of them is have a very high dimension, so it can have more than 100,000 of columns. And also it's extremely sparse. So actually most of the entries, and this is really independent of the protocol or the data that you look, you're going to see that at least 95% of these entries are going to be zeros. And we believe, or it's easy to show actually that some proportion of these zeros are possibly dropouts, which are coming from the fact that in your single cell toxic experiment, you're going to have material losses. So as an example how this happens, if you go back to the toxic protocol, actually to get a valid fragment, you need 2 transposition events. And the TN5 actually has 2 different adapters, but only fragments that have 2 different adapters will be actually amplifying the PCR. And this only happens 50% of the time. So only this step, half of your DNA is getting lost. And assuming, for example, the further half of your DNA will be lost, then it's easy to show actually with simple math and simple assumption that half of your open chromatin regions are going to be lost actually in your matrix. So this leads us to try to propose methods to, let's say, denoise these matrices. And the method is based on positive and labeled learning. And what the method does is that it receives a binarized matrix. So we are now only assuming there is a read or not, and trying to define probability that one particular region is open in one particular cell. And this matrix we can actually use as input of any kind of further downstream methods. So how the method works? So we want to estimate this matrix M. And this is now given the probability that your chromatin is open or not, right? And this is decoded by this matrix Y. And we can encode this using this easy probability rules. The thing is that actually we don't really observe Y. We observe X, which is a matrix which has dropouts, right? So some of the ones, for example, here, are likely to be lost because of loss of DNA material. So what we do now is actually we can try now to estimate the probability on this matrix observed matrix X. And this is now related to, but to account for the dropouts, we put a term here, which the higher it will be, then the higher will be the probability to have a dropout. And we encode this term in a way such that it's cell-specific and should be also proportional to the read coverage in that cell. So cells with have higher coverage should have a lower dropout, or the way around. So we can then use now positive and label methods to solve this problem. And we actually use a constraint optimization problem described in this paper. But we actually need to improve it, for example, to make it applicable to single cell data. For example, we relax constraints to make it that we can estimate the matrix inefficient, so in a few minutes, and also to allow this cell-specific dropout modeling. So how do we evaluate this? And for example, we use this data set from Buenarosa et al., where a single cell taxic was performed in pre-sorted cells. We mix the cells, so we don't give this information to the methods, and we evaluated different imputation methods, and for example, and then perform clustering. And as you can observe here, for example, in the t-SNE level, SC-open does a quite good job in separating all the different cell types. Other methods, for example, cystopic, also do quite well. But if, for example, we try to measure the accuracy in the clustering level, we see that SC-open has an advantage. And actually, we look at all the benchmarking data sets, and we always have this kind of picture. We also evaluated SC-open also before using different methods as ChromaVar, and we could always see that imputing with SC-open would always improve. We can see more details in the print below. So the next part is actually going to show how we can use footprinting in the single cell taxic data, and here to try to study complex disease. And here we are interested in fibrosis, which is this process where we have hardening of a tissue, in this case here, the kidney, and this is because we have accumulation of matrices. And this process is in the cellular level happens that, let's say, due to your disease, you're going to have injured cells, for example, this injured paroxysmal tubular cells, and they are going to trigger some signaling. For example, they will activate fibroblasts into matrix depositing myofibroblasts through, for example, TGF-beta signaling. But actually, the crosstalk is more complicated, involves also very different cells, and we don't have a fully understanding of how this is working. So we wanted to understand what a regulatory change is here, and for this we use a mouse model where we have kidney injury, but this also produces fibrosis, and we measure this mouse 2 days and 10 days after this obstruction. And we could recover 25,000 cells, and we could also characterize more than 14 kidney cell types. First thing we asked was, okay, can we see changes in, for example, cellular counts, and what was quite interesting to see was that actually we see, for example, loss of proximal tubular cells expected, but also increase of these injured proximal tubular cells. We also find the subpopulation of fibroblasts, which were increasing over time, and these we characterize as myofibroblasts. And we also saw other things which are hallmarks of this model, for example, loss of endothelial cells or increase of immune cells. Okay, but of course we wanted to understand more, not only to do cell counting. And to do this, we use footprinting analysis to try to predict transcription factor activity in individual cell types. And as you can see here, for example, was quite interesting that we see that many factors highlighted here and are shared across all the proximal tubular cells. But interestingly, for the injured proximal tubular cells, these factors have low activity. And this is exemplified here by this RxRA, where you see that in the injury case, it's much lower than in the healthy or in the normal proximal tubular cells. Here, actually, we could even further down and look at the changes over time. So this is, and you can see that actually the change of this particular factor it was actually happening over time in a stepwise manner. So this kind of indicated this injured PT, so proximal tubular cells, they kind of de-differentiate, or they lose their identity by having low activity of the factors which are usually defined proximal tubular cells. Other thing that we found was quite interesting was this, that the myofibroblasts, they have very different activity compared to the fibroblast cells, but also they share actually this activity with injured PT cells, indicating that both cells are kind of regulated by the same inflammatory programs, and they are associated to factors like, say, as BTF2, but also SMAD factors. So actually, SMAD factors are actually associated with TGF-beta signaling, and as you see, they are quite high in both injured proximal tubular cells, but also myofibroblasts. Also interestingly, if you look, for example, at the activity of this factor over time, we see that at day 2, it was already quite high, indicating this was already a very early response to this kidney injury. So as I show here, that actually we can use transcription factor activity to characterize the regulatory programs which are governing these different cell types. And so I hope I have convinced you that as the single cell toxic data has this issue with the data sparsity, and the imputation, in particular using SCOpen, can address this problem. And also that we can use footprinting analysis to dissect complex processes, as here shown in these temporal changes during kidney fibrosis. And if you are curious to see more results, for example, we also have further characterized, for example, transcription factors which are driving this fibroblast to myofibroblast differentiation, and also potential target genes and potential target sites. So meaning that we can really make use of the single cell data to really characterize changes at these subcellular levels. With this, I'd like to thank you for your attention, and also Jinjian Lin, which actually was doing most of the work on the computational side here, and our collaborators, Rafael Kramer and Christoph Cooper, who did most of the wet lab work used here. Okay, and thank you for your attention.
Thanks, Ethan, for the very interesting talk. So, audience, if you have questions, please type them into the chat box. Okay, so we have a question from Ziqi Zhang. The question is, may I know you estimate the sampling rate dropout probability from the data set? I guess, like, may I know how you estimate the sampling rate or dropout probability from the data set in SCOpen? Also, a problem about the method part is the final output of SCOpen, which is visualized in t-SNE, the probability matrix M, or do you do some post-processing to obtain the binary matrix?
Sure. Yeah, thanks for the talk, for the question. I mean, concerning the dropout, I mean, you can see more details in the biotype, the preprint, but the idea is that we look at the distributions of fragments per cell, and we have a formula which is based on where we have an assumption, which is kind of the amount of reads will be then in the log scale, reducing the dropout rate. And but this is something that actually we are currently working on, because it's a parametric method, and we are trying to improve it in a way that makes it non-parametric, and hopefully you'll see soon a new version of the method where actually this is being estimated directly from the data from the data in non-parametric way. Concerning the second question, can you maybe repeat this, the second part?
Yeah, the second question is the final output of SCOpen. So it's this is refers to the visualization that you did in TISNE. So
okay.
Right. So is that the probability matrix M, or is that some post-processing procedure to obtain the binary matrix?
Yeah, so the output is, the final output will be this probability matrix. So we're going to be values from 0 to 1. And out of this matrix, then you can input any kind of downstream methods. It can be TISNE or DoMap, or we actually have tests that you can also check the preprint. For example, also as input as ChromVar, or even systopic. And what we see is that whenever you do the imputation, we improve clustering, but also other type of tasks, which are, let's say, downstream of the imputation.
Okay, thanks, Ivan. So we have time for one more question. This is a question from Ansho. Very cool model. Do all the TFs show case sufficiently detectable footprints in single-cell ataxic or ataxic profiles? So there is actually a second question, second part of this question. Or do you think footprints are biased towards TFs with higher residence times or other properties?
Yeah, I think this is again a very good question, and I mean, and I think there is actually literature also from my group, which you have to follow also through the when we are doing footprints of the bulk attack. And what we see is that indeed not all the factors actually have nice footprints, and also the residence time is something that's relevant. So I would say 2/3 or maybe half of the factors we see nice footprints. And of course, the ones that we are showing and that we are highlighting, because the TF activity score, which we show, are based on the fact that these factors have nice footprint, then they're going to be the ones that these nice footprints are coming up. And I think, of course, this is a limitation of using DNA footprint and either attack DNAs, but that's the price that you have to pay when you're using such a protocol.
Okay, thanks, Ivan. So I think we should move to the next talk.